والصلاة والسلام على سيدنا محمد وعلى آله وصحبه أجمعين. My name is Dr.Sara Ahmed. Today we are going to have an interesting topic. I hope that you will enjoy it. The topic is called normal flora. It's part of a series of lectures that we have been presenting here to help our daughters and sons, أبنائنا وبناتنا, who war deprived them from having the regular academic life, but this is actually a hope in life. Never lose hope, وإن شاء الله تستمتعوا بالمحاضرة بإذن الله تعالى. طيب. Let me just check why it's not next. So what is expected in this lecture? That we will define some terms: normal flora, resident flora, transient flora, and carrier state. We also will know the origin of normal flora. We also will understand some normal flora examples, considering the source of opportunistic infections, autoimmune stimulation, some benefits of this normal flora: nutrition, vitamins, production of carcinogens, etc. And also we will know the residing bacteria on our body, where they are, what types are there, and to know that no one is sterile from normal flora. Normal flora is there. It is part of us. Okay, bacteria are telling you that we are not all bad. مش كل المايكرو اورجانيزمز اللي موجودة على جسمنا معناها إن هي عاملة infection. It is causing an infection, and we are supposed to take antibiotics to deal with them. So it means that if you have a microorganism in or on your body, it doesn't mean that you have an infection. But we need to understand, or we need to define this presence and to understand which is it normally there, or is it a pathogenic organism or something like that. We will talk during this lecture. We will understand about this more, inshallah. طيب, some definitions. A normal flora are microorganisms that are frequently found in a particular site in normal healthy individuals. So it is fascinating to know that normal individuals, they have actually bacteria on or in their bodies. But the thing is that their presence there is actually a good thing, and we are going to understand why it is good in the coming few slides, inshallah. Some are found in association with humans or animals only. So having a normal flora, having microorganisms residing on our bodies, not causing infection but benefiting mutually from each other, could be unique, so that the composition of the microorganisms present on the skin is not like the organisms that are present on or in the vagina, or present on the conjunctiva in the eye, or, for example, present somewhere else. And usually when we talk about normal flora, or organisms residing on our body normally or naturally, we are majorly talking about bacteria. It doesn't mean that bacteria only are the part of the normal flora. For example, in the colon, we have parasites that are actually present in our colon, but they are not causing us any kinds of infections. But the majority of the normal flora are actually bacteria. There is a symbolic relationship with the host. It means that there's a sharing benefit. You provide them with the place to live, and they provide you with some nutrients that your body requires. You provide them with a nutrients, and they provide you with protection from being colonized with very dangerous organisms. يعني هم حرفيا كده بيكونوا موجودين مثلا على السكين في شكل زي البساط، مغطيين السكين، مش سامحين لأي أورجانيزم تاني إنه هو يجي ويكون ريسايدنج على السكين بالإنابة عنه. فبيكون مغطيك، مقفل المكان تمام، بحيث إنك أنت ما تيجي حاجة ممكن في يوم من الأيام تعمل لك مشكلة، ولا تعمل infection ولا يكون primary pathogen ولا يكون drug resistant organism. So the relationship with the bacteria or with the normal flora is a symbolic relationship. Type, it is subjected to constant changes. So it means that this normal flora is not static since birth and until the person becomes an adult or an older adult. It is subjected to change because we take antibiotics during our lives due to different causes. So it means that we sometimes kill some of this normal flora. So other organisms might come, or some normal flora might be more in number than other flora and whatever. Or sometimes during your life, you actually meet someone who is a carrier for example, Neisseria meningitis, and you get colonized in some part of your life with Neisseria meningitis. You don't have the infection, but it is there. So you acquired it during your life, and etc. So it is not a constant environment. The number of the bacteria are preserved and they never change. No, it is actually subjected to constant change due to different environmental factors, due to host factors, due to nutritional factors. You might change the normal flora of your colon through the way that you are eating. Also altered by antimicrobial agents. So antibiotics, as I mentioned before, will change somehow the normal flora, because if you take an antibiotic, which is an antibacterial, for some reason, it will make a change in your bowel normal flora in the colon, which requires around 2 years of life to get back to the normal situation that was before. So taking an antibiotic might treat an infection, but taking an unnecessary antibiotic may disturb the normal flora of your colon, for example. Okay? Okay, the types of normal flora, we have commensals. When we say commensals, it means that microorganisms that have a natural relationship with the host, the human being. So the commensal has a natural relationship, has a relationship that is not described as an infection. There are no pathogenic consequences on the host, and the organisms are there benefiting and also providing us with benefits. Residents, present for a constant period of time. They are regularly found in a given area at invariable period, and if they are disturbed, they promptly re-establish themselves. So the residents are actually the normal flora that is staying and not changing. Even if something like an antibiotic was taken, changed the resident bacteria, after a while they will regain the same establishment as before again. For example, after stopping the antibiotic. Transients, which are non-pathogenic or potential pathogenic. So the transients are not all of them normal flora. Sometimes some pathogenic organisms may transiently colonize, for example, Neisseria meningitis colonizing the throat. So that they inhibit the skin or mucous membranes for hours or days. So they are there and they are present for hours or for days or for a period of time. They establish itself briefly, excluded by the host defense or competition from residents. So when the resident flora finds that there is a bacteria that is strange to it, they will try to compete with it to eliminate this strange bacteria or this pathogenic organism. Or this pathogenic organism, finds its way to cause infection or disease. So either the host immune response will get rid of this organism, or the competitive flora will compete with it for nutrients and compete with it for the space that it is occupying, or the person might get infected eventually by this organism due to any cause. So commensals, residents, transients, and then there is what is called a carrier state. So a carrier state is actually potentially pathogenic. So it means that these organisms, they are pathogenic, but the body couldn't get rid of them. They are staying there for a long time. And actually, like Neisseria meningitis, they were found that they are colonizing 10%. So if the normal flora here, the resident flora, couldn't get rid of these transients, and it continued for a longer time, it means that in this situation, we will call it a carrier state of microorganisms. It's like Streptococcus pneumoniae, which is everyone has Streptococcus pneumoniae in the upper respiratory tract. Neisseria meningitis, 10% of people might have Neisseria meningitis in their throat, but due to the vaccination now, these things are much more better. So, if we talk about from where did the first microorganisms colonizing our body come from? The newborn, when the newborn is born through the genital tract of the mother, is usually sterile. The uterus is sterile, so by any means, there is no mean that this newborn is sterile in utero, not sterile. But when it is born, after birth, it is exposed to the normal flora of the mother's genital tract. Please close the microphone. Close the microphone. Okay. The normal flora of the mother's genital tract, skin, respiratory tract of those handling him, and the organisms in their environment. Now we understood how do we get our normal flora. During the birth, while it is getting out of the genital tract of the mother, it will acquire the normal flora from the genital tract and also in the respiratory tract as well. And then the people who are handling this child, and also from the environment, because the child starts to get in contact with the bed linings, with the people arms, they are kissing and even carrying them, they are bathing them, the water organisms, and etc. Okay? You can see here in this diagram, during pregnancy, the mother normal flora itself is affected by diet, antibiotics, host genetics, infection, and maternal stress. And during delivery, according to the mode of delivery, the organisms that will colonize the infant will change. Also, the gestational age of the baby, when it is what was born, whether it is a preterm or it is a full-term baby, and also the vertical transmission from the mother to the child. This changes the normal flora of the infant or the child. So, infant, the window of opportunity for microbiota starts from here after delivery, from infancy to toddler, and also in the toddlers it is affected by the feeding mode of the child, whether he is completely breastfeeding or he is on bottle feeding and breastfeeding or only bottle feeding. The maternal diet, the mother is providing him with diet. She is making meals for the child according to what she knows or what she read about. The host genetics themselves, the family lifestyle, and the geographical location, because also there we have different organisms that might reside a different population in a different area. For example, the people who are in the mountains or the people who are living far in the urbans is different from the rural, and etc. Okay, bacterial beneficial effects of normal flora. What are the benefits from having this normal flora? Are they just residing there just to compete with other organisms, or there are other benefits? Actually, yes, there is. The first one is immune stimulation. Actually, because these organisms are present on the skin and also on the mucous membranes, for example, in the upper respiratory tract, in the genital tract. They are present in the colon, which also has its mucous membranes. So there is an interaction with the immunology of the body. And hence, when the immune system, for example, like the IgA antibodies, gets encountered with these organisms, or the IgG there, or being opsonized by some immunology cells inside in the body, Then the antigens of this bacteria will be presented, and then you will have a population of immune cells that are knowledgeable about this organism. So definitely this is called immunostimulation. Also, there is exclusionary effect, which is the vacuum effect, and protection from external invaders. As we mentioned, that when they are there, no other bacteria will colonize the area that the normal flora is present in. And again, the production of essential nutrients, for example, vitamin K and B, by some normal flora of the intestine, for example, E. coli. It's a fecal flora, but it produces for us vitamin K and B, and these are important vitamins for our body. If you can see here, you will find that these are the organisms that are present in the colon, and these are the columnar epithelium of the colon, and here are the M cells. And these organisms, when they counter with the immune system, you can see that there is a cell-mediated, there is a cellular immunity here, and that the eventual, what happens is that there is an IgA that is comes from the proliferation and differentiation of these immune cells after getting encounter with the antigens of the bacteria, and after they are presented through the antigen-presenting cells, for example, macrophages. So we have cells with memory cells and production of antibodies that are specific antibodies. So there is immune stimulation on this area. And unusually colonized intestines, like in the cesarean section, non-breastfeeding, or the use of antibiotics. So this is actually unusually colonized intestine, but normally colonized intestine usually happens in natural delivery and after breastfeeding. So you will have the usual or the normal combination of organisms that depends on several factors, as we mentioned before, and also on this slide. So bacteria, we have protection. The commensal organisms will enhance the formation of the inflammasome, and then the migration of dendritic cells, for example, from the lungs to the lymphatic tract. And here you can see that there is this influenza virus where these cells are migrating there and can eliminate the infection by viruses which could be dangerous viruses. Indirect promotion, commensal bacteria also sometimes they enhance the lymphoid cell proliferation, and direct promotion, some of these cells might affect directly and give us an antiviral immune response due to this commensal reactive. Okay, actually here you can see that metabolites production in the liver, vitamin K production, and eventually structural modification of VKS molecules. You can see that these are the gut bacteria. You can see the bacteria there, and you can see how they benefit and what exactly they do, and some drugs that might inhibit that. Other factors regarding normal flora, it may be a source of opportunistic infection. So let me just tell you what is opportunistic infection. Opportunistic infection, it means that there is an infection by an organism that is not usually causing disease in the human being or in the body. And we have, if a normal flora, which is residing, for example, on the mucous membranes of the, for example, of the colon, finds its way from the area that is usually present in to an area that is usually sterile or sterile. If it finds its way inside this sterile environment, then it causes infection, even if it is a commensal, and even if it is beneficial in the area that it is colonizing. Another type of opportunistic infection is that if the organism is not known to be part of the normal flora of the body at all. For example, Staphylococcus saprophyticus, which is a saprophyte organism present in the soil. It can cause urinary tract infection in the humans because it comes from the outside environment, which is its original environment, and then it finds its way to the urinary tract, for example, of the females, and it causes urinary tract infection. So also this is an example of an opportunistic infection. So for an opportunistic infection to happen, either the area is sterile, no organisms is there, and a commensal organism finds its way to this sterile environment. This is an opportunistic infection. Or the organism is not known to cause disease and not colonizing the body at all, but it finds its way due to one factor or another. So in patients with impaired defense, and the third one also for opportunistic infections, that there are colonizers which are colonizing, and the immune system knows about them, and there is this boundary that the colonizers doesn't cross. But what if this person or this individual has a lower state of immune defense? He doesn't have his immune system is compromised. So these organisms that are residing as colonizers will find its way, and it will cause infection somewhere else in the body. This is another type of opportunistic infection. Now we have three types of opportunistic infection, and the latest one is that when the immune system of the host is compromised. Some may cross-react with normal tissue components. For example, some organisms, they have antigens which are present on their cell wall that might cross-react with some antibodies and will cause to have an antibodies, for example, for the ABO system, the ABO system. And because of the cross-reactivity between the intestinal flora and the antigens of the A and B blood substances, antibodies develop to the ABO system. You might ask me how the antibodies of the ABO system develop, because we have mentioned that the infant is coming from a sterile environment, which is the uterus. So how, if I want to give blood to a neonate, I will find out if he has the B group, he will have antibodies against the A. From where did this come, these antibodies? Actually because of the normal flora, because these antibodies are developed due to the antigens present in bacteria, which cross-reacts with the ABO system. I hope this is clear. You can see that ABO antibodies, we have natural occurring antibodies for the ABO groups, develop as bacterial normal flora populates the gastrointestinal tract during infancy. They have a thermal range of 20 to 33 degrees centigrade, and they are of the IgG or IgM type ABO antibodies. Also, there is the production of carcinogens. Production of carcinogens might be due to some normal flora modifying through their enzymes, modifying chemicals that is present in our diet, for example, artificial sweeteners. So by enzymatic change of certain bacteria, for example, E. coli, there might be presence of bladder carcinogens, and by hence they are causing bladder cancer. When we come and talk about the distribution of normal flora, we'll find that in the internal organs, except the alimentary tract, all of the internal organs are sterile at health. Sterility is maintained by the local defense mechanisms. These organs are sterile because their local defense mechanisms, the chemical substances in serum and tissues, for example, complement and antibodies, and also the phagocytic activity of the polymorph nucleosides, the PMNs or the neutrophils. All of these factors, they maintain our internal bodies sterile, free from normal flora. Usually what I say is that any place in the body that is exposed to external environment, you will find normal flora in it or on it. So it means that the name internal organs, it means that it is not exposed to the outside environment. So this is why it is sterile. So the nose is colonized, the mouth is colonized until the anterior part or anterior some part of the nasopharynx. And then when you go down to the trachea and the bronchi in the lungs, you will find that these areas are sterile. Okay. Also, we have the areas here that are normally having or harboring organisms. The skin, number one. We have in the GIT the mouth and the large colon. And in the urogenital tract, we have the vagina and the distal third of the urethra. So the anterior third of the urethra, so the distal third of the urethra is actually harboring the skin organisms which are present in the perineum area. And you will find that there are microorganisms. This is why when we want to collect urine for culture and sensitivity to detect what bacteria causing urinary tract infection, and because urine usually passes through the urethra in males which is conjointly urogenital system, and in females also passing through the area near the vagina. In the anterior third of the urethra, we ask the patient to pass the midstream urine, so that to wash out the anterior third colonizers and give us the midstream urine, so that we can find what organisms are inside the bladder actually causing an urinary tract infection. So when you know about the normal flora, then you understand what types of samples you want to diagnose a true or a real infection. So now you can see distribution of the intestine flora. You will find that in the intestine, the predominant organisms and the concentration per gram. Actually, in the colon, the stool, the organisms are weighed by grams. So in the stool, we'll find obligate anaerobes like streptococci, staphylococci, and Neisseria, which is more than 10 to the power 6 per gram. And lactobacilli, streptococci, less than 10 to the power 4. Anaerobes like Bacteroides, coliforms like E. coli, streptococci, and Candida and protozoa is actually 10 to the power 11. If you can just follow me, the darker shade is here. You can find here, these are the place for anaerobes. This is the area of anaerobes here. The lower part of this table is present here in this area. And the lighter one, the lactobacilli and streptococci, is here. And here is no organisms because it's the nearest to the small intestine. And we have here the obligate anaerobes might be here. So the deeper you go, you will find the aerobes are there. And when you are coming to the outside, this is the anal orifice, you will find that there are multiple organisms. And as we mentioned before, that bacteria are not the only colonizers or the only commensals. Also we have protozoa here, Entamoeba coli, Dientamoeba fragilis, they are all present here, and Candida, which is a fungus. Normal flora of the respiratory tract, the upper respiratory tract is colonized by flora as in the mouth and nasopharynx. The lower respiratory tract is sterile or sterile. The nose, there is Staphylococcus epidermidis, Staphylococcus aureus, and Corynebacterium other than Corynebacterium diphtheria, other. Oropharynx, we have viridans streptococci, commensal Neisseria, Corynebacteria, Bacteroides, and Fusobacterium, Veillonella, Actinomyces, Spirochetes, and also Haemophilus influenzae, Pneumococcus are all. These are potential pathogens in this area, and less commonly Streptococcus pyogenes and Neisseria meningitis. So the least the organism is about to be found, the more potentiality to cause infection due to one cause or another. So for Haemophilus influenzae and for pneumococci, they are actually potential pathogens. If it may colonize the oropharynx, and the least common to be present is Streptococcus pyogenes and Neisseria meningitis, and for Neisseria is the least because due to vaccination and other causes. Gastrointestinal tract, we have saliva, which contains 10 to the power 8 bacteria per ml. If you tell someone that his mouth contains this large number of organisms, they will not believe in regular human beings, I mean. But you need to understand that our mouth is not sterile. We have in the saliva, 1 per ml, we have 10 to the power 8 bacteria. So the bacteria continually colonize gingival margin debris and dental plaque, and also there is esophagus flora as the same as the pharyngeal flora. An empty stomach is sterile due to the gastric acid. And this is a way of Allah's creation to not to harbor any organisms when the stomach is empty. And of course, there are infections that can happen in the stomach, for example, Helicobacter pylori that causes stomach ulcer or peptic ulcer, but usually the stomach is sterile. So the presence of any organism in the stomach means that this is an abnormal situation. Duodenum and jejunum and upper ileum have scanty flora, and the large intestine is heavily colonized by bacteria. The feces. 1/3 of feces weight is bacteria, mainly dead bacteria, and some are living, which are approximately 10 to the power 10 per gram, and 99% are anaerobic bacteria. So the anaerobic environment maintained by aerobic bacteria utilizing free oxygen. So how they survive the anaerobes? Why are they 99%? Because the aerobic bacteria is living there, and actually they are utilizing the free oxygen. So it makes the situation much more anaerobic, and then the anaerobes, they increase in number. Bacteroides fragilis group is the dominant anaerobe. There is also bifidobacterium and lactobacilli. Less commonly is E. coli and Proteus, but E. coli is the commonest of the facultative anaerobes that are present in feces. Is the commonest. Distribution and frequency of bacteria in the intestine. Here's the density and here's the frequency of occurrence. The esophagus and the stomach, they might be, the esophagus is colonized by lactobacilli, which is a gram-positive anaerobic bacteria, bacilli. In the small bowel, the duodenum, and the ileum, we have the small bowel here, might be colonized transiently by lactobacilli streptococci. And the more you go through towards the colon, you will find the enteric bacteria and some anaerobes. For the large bowel, you will have Bacteroides species, which is Bacterium Enterococcus fecalis, which is a gram-positive streptococci, E. coli, and other Enterobacteriaceae like Klebsiella and Eubacterium, Bifidobacterium also. Also, there are Lactobacillus, which is a gram-positive bacilli, Staph aureus, Clostridium species, and also streptococci like Pseudomonas and Salmonella. Salmonella is a primary pathogen, it colonizes, and Pseudomonas is also a transient colonizer. And fecal material contains the anaerobes mainly, which is the Bacteroides species, and other coliforms, and also Enterococcus fecalis. You will find the lecture in the Moodle there. You can take a look again. Genital flora. The female genital tract is heavily colonized. If we ask ourselves why, hormones changes the normal flora. The hormones, whether the hormones during the life, the fertile female, during having her periods, she's alternating between having a period and not having a period, and this changes the normal flora of the vagina. Also the dietary habits. If they are taking a lot of carbohydrates, it will enhance the presence of other organisms than the organisms desired, for example, like Candida, and then what happens is that it changes or alters. Habits like the washing habits or the hygiene. Also, after delivery, there is a change, of course, and the vitamins in the body, there's a change in the immune defense, and then the change in the normal flora as well. The relationship between the microorganisms in the genital tract is actually commensal, symbiotic, and there are some pathogenic organisms that inhabit the vaginal surface and its cavity. Around 10 to the power 8 ml per ml in normal vaginal secretion, there are bacteria. And in both sexes, we'll find Mycobacterium smegmatis, which is an acid-fast bacilli in the secretions that might sometimes contaminate urine, leading to confusion or misdiagnosis. Mycobacterium smegmatis, this is actually the organism that is found in the frenulum of the external genitalia of the male, and it is actually decreased or even by the circumcision in males. And it is actually the presence of Mycobacterium smegmatis in males in the frenulum. It actually is related with the cancer of that area. So actually circumcision in males is beneficial due to, this is one of the causes. Male and female distal urethra, they have Staphylococcus epidermidis, Corynebacterium, and also Mycoplasma, which is a cell wall free bacteria. It's an atypical bacteria. The female vulva, the external genitalia, Staphylococcus epidermidis, as we can see that epidermidis from the name, epi is dermis, epidermidis is above the skin, dermis the skin, so this organism is found on the skin. Corynebacterium and E. coli and other coliforms. You can see that the vulva is actually a mixture between the gram-positive organisms that are present or residing in the skin and the organisms of the colon. So the perineal area is actually a place for organisms that are part of the normal flora of feces. Vagina, there is lactobacilli, which is called the Doderines bacilli, Bacteroides, also Enterococci, and Corini, Mycoplasma, and some yeasts. These are residing there normally. You can see, can you see your hand now? This is actually a hand cultured on a petri dish. You can see how many the organisms are residing on our skin. Am I right? So the skin is rich in bacterial flora, definitely, and it is actually 10 to the power 4 per cubic centimeter. And they exist in the form of microcolonies, and you can see here how it looks like. You can see here, this is a culture of a hand. This is the hand. Yeah, it's fascinating actually. How many organisms do we have? Main skin flora: Propionibacterium acani, which is the causative agent of the infected acne. Most of the people, they are suffering from anaerobic cocci. It's a skin, yeah, there is an anaerobic cocci inside the sebaceous glands. Staphylococcus epidermidis, Corynebacterium, Staph aureus, which is a less common and a potential pathogen, also coliforms. The commensals on the skin help to break down the dying skin cells or destroy the debris secreted by many minute glands and pores and open on the skin. External auditory mediators: Staphylococcus epidermidis, Corynebacterium, and acid-fast bacilli occasionally might be in the wax. And the conjunctival sac flora, there is Corynebacterium xerosis and Staphylococcus epidermidis, also Myxella catarrhalis might be one of the commensals. The summary is that not all bacteria in our body are bad. We need to know the different bacteria residing in our body, and the newborn harbors bacteria for the first time from their mother and the external environment, and colonized by pathogenic bacteria is not uncommon. Now these are some of the lectures here, some of the references, and I hope that you have enjoyed the lecture. If you have any questions, please ask. Thank you. If you have any lectures, if you have any questions, anyone? في حد عنده سؤال؟ عائشة، لنا، يسرى، أروى، سحر، إسراء.
دكتور
جزاكم الله خير. الشباب الباقيين عندكم أي حاجة؟
يعطيك العافية دكتور.
الله يسلمكم. المحاضرة هتلقوها في المودل إن شاء الله. feel free إنكم تمشوا تشوفوها مرة ثانية، هي interesting وظريفة إن شاء الله، وأتمنى لكم كل التوفيق يا أولادي.
شكرا يا دكتور.
مع السلامة، شكرا جزيلا.